Welcome all to the today's session. So today we are going to discuss the structure of DNA. Okay, this is very important topic from the examination point of view. Certain things we have already seen about the purine and pyrimidines, etc. In the last session. So let us talk about the DNA. Okay, so we have seen the basic component required are the sugar, which is deoxyribose. Then base is A, adenine, guanine, cytosine, or thymine in the DNA. Okay. In the RNA, T is replaced by U. That also we have seen. The bond is a 3'5' phosphoesterase bond. Okay. So how the nucleotide is formed? We have seen it will form from the base plus sugar plus phosphate. That will form the nucleotide. And nucleoside is a base plus sugar. Okay. So we know from our basic knowledge there is a Watson-Crick model for the DNA. Okay. So if you see, there are two strands which are arranged in a helical format. Okay, these two strands are attached to each other by the bonds. Okay, so there are bonds and there are bases which are attached to each other. Okay, so the key structure is that it is a right-handed double helix. Strand is oriented anti-parallel. That means one strand is in the 5'3' direction, other strand is in the 3'5' direction. Okay, the base adenine will combine with thymine and guanosine will combine with the cytosine. Okay, so strand orientation is anti-parallel. One is in the 5'3' direction and other is a 3'5' direction. Base pairing is A will combine with T and there is a double bond between the two, and G will combine with C. Guanosine will combine with the cytosine and there is a triple bond. Okay, and this is a very high yield bond or very stable bond. This triple bond is a very, so if you have a DNA which is rich in guanine and cytosine, it is more stable because it has got a 3 hydrogen bonds. Okay, so there is a Chargaff rule which says that adenine will combine with thymine and guanosine will combine with the cytosine. Okay, and the polarity which is written already is from 5' to 3'. Okay, now let us see chromosome and the DNA organization. So DNA is a double helix. Nucleosome is a DNA plus histone octamer. Chromatin is a condensed nucleosome and chromosome is a highly condensed DNA. Okay, so when you have the DNA, the structure is a double helix. Nucleosome will have the DNA plus histone octamer, which we'll see in the figure now. Okay, so there are some histones. Okay, there are some core histones and the linker histone. The core histones are the H2, H3 and H4. H2 is H2A and H2B and H3 and H4, and linker is only one, that is H1. Okay, so this is what is the octamer core. Okay, 1, 2, 3, 4 and similarly 5, 6, 7, 8. Okay, so these 4 plus there are 2 here and 2 on this side. So this is the octamer core of the histones. Okay, and these core ones are the, there will be 2 H2A, there will be 2 H2B, there will be 2 H3 and there will be 2 H4. And there is a one linker histone. Here is a linker histone, that is H1. Okay, so you have got a linker histone which is H1. Okay, so this is how you get a nucleosome. Okay, so there are two procedure. One is acetylation. Once there is a acetylation, it will increase the transcription, and there is a methylation that will reduce the transcription. So if there is a acetylation, it will lead to increase in transcription. If there is a methylation, it will lead to decrease in transcription. Okay, what happens with acetylation? The effect on chromatin that it becomes open, so the transcription is increased. What happens with methylation? The chromatin is closed and there is a decrease in the transcription. Okay, so chromatin is of two types: euchromatin, which is active, and heterochromatin, which is inactive. Okay, now coming to the DNA replication, there can be question which is asked on this. So this DNA replication happens in a semi-conservative replication. Each daughter cell will get one parental strand and one new strand. We have seen this DNA structure is in helical format. So when there is a transcription, these two bands will separate. So this strand will separate and this strand will separate, and another new strand will form for this, and another strand will be formed from this. Okay, we know that there is a one 5'N and other is 3'N. For the other strand, it will be 3'N and this will be the 5'N. Okay, so these are the certain things which you should remember. Okay, So the basic principle is that it is semi-conservative replication. That means each daughter cell will get one parental strand and one new strand. That is what is the nature semi-conservative replication. So what are the steps? The first step is the initiation. That is origin of replication. It begins at origin of replication, and there are ORC proteins which are responsible for binding. Okay. And next step is unwinding. The first is a initiation. Second is a unwinding. For unwinding, three enzymes are responsible. One is a helicase, which will cause unwinding of DNA. Then there is a topoisomerase, which will relieve the tension or the supercoiling. And then there is a SSB, that is a single strand binding proteins that will stabilize each bond. Okay. So if you see here in this figure, so this is the double helix. Okay. One is a 5' end, the other strand is of 3' end. Same here for this 3', if you see the end is here is a 5', and for this strand the other end is a 3'. Okay, so this is the parental double DNA double helix. Okay, now there has to be replication of this. So first there should be the unwinding of this. Okay, so if you see here, this is the unwinding of the parental DNA. Okay, so the unwinding of the DNA, the enzyme is a helicase. Okay, it unwinds, and then there is a SSB proteins. Okay, we have seen helicase, SSB, and topoisomerase. Okay, helicase is responsible for unwinding the DNA, while SSB is responsible for stabilizing the individual strand. Okay, so this SSB will stabilize the individual strand. Okay, and then comes the role of the DNA polymerase. Okay, so that we will see subsequently. The next step is the elongation. The elongation will happen in the 5' and 3' direction only. Here you should remember that elongation happens in the 5' and 3' direction, while the template is read in the 3' and 5' direction. Okay, see here it is a 5' end, and the same strand other end is a 3' end. So this is 5' and this is the 3'. This is the only one strand which is there. Okay, so this is the strand which is there, and this is the 3' end, and this is the 5' end of the other strand. Okay, now you see here, suppose this is the 5' end and this is the 3' end. The other strand will have 3' end here, and here will be the 5' end. Now after separation, this will get separated from this. There will be new strand which is formed here. There will be new strand which is formed for here, and you will have two strands separately. Okay, now this strand formation always takes place from 5' to the 3'. So the reading, now here it is a 5', here it is a 3', but the strand will get formed from the 5' to 3' direction. So there is a reading of the codon from 3' end. So here for 3', the end will be 5' here. Okay, so the codon is read, or the proofreading is done from 3' end, and the synthesis begins from 5' to 3' end. Okay, that is what you should remember. Okay, so the direction is always 5' to 3', and template is read from 3' to 5'. So once the template is read, it will form the complementary band, and the complementary band direction will be 5' to 3'. Okay, so reading of the template is in the 3' to 5' direction, but the band gets formed in the 3' and 5' direction only. So DNA replications starts at origin. Helicase will unwind the DNA, forming a fork. Then primase lays the DNA primer. DNA polymerase synthesizes the new strand in the 5' 3' direction. Remember, DNA polymerase cannot act alone. It requires a primase. Okay, so this primase lays the RNA primer, and then the DNA polymerase will act, and it will synthesize the new strand from 5' to 3' direction. There is a leading strand which is continuous. Lagging strand forms the Okazaki fragments, which are later joined by the ligase. Replication is semi-conservative. So first thing what should happen, these two strands should get separated. So these two strands should get separated. So there is a helicase enzyme. Then there is a reading of the codons. Reading will start from the 3' to 5' end, while synthesis will begin from the 5' to 3' end only. Okay, and then there is a leading strand, and then there is a lagging strand. Okay, for leading strand, what is there? For leading strand, it is in a continuous manner. The activity is in the continuous manner, while lagging strand, there is a discontinuous manner, and the lagging strand will have the Okazaki fragments. Okay, so there are different DNA polymerases, that is alpha, beta, gamma, epsilon, delta, etc. So key point is that it requires a RNA primer. Okay, this DNA polymerase cannot act, it requires RNA polymerase. It should have the first proofreading, which is in the direction of 3' to 5' exonucleases. So DNA polymerase cannot start synthesis on its own. It can only add nucleotides to an existing 3-hydroxyl end. Okay, so DNA polymerase needs RNA primer and has 3' 5' proofreading activity. So it has got activity of proofreading in the direction of 3' to 5', and that strand will get formed in the 5' to 3' direction. Okay, so DNA polymerase checks and corrects errors during the replication. Okay, so RNA primer initiates DNA synthesis. Proofreading prevents the mutations. Okay, then Okazaki fragments. This is in the lagging strand. Okay, the lagging strand is in a discontinuous format. It will form the Okazaki fragments. So the strand is a lagging strand. Nature is a discontinuous, and size is 100 to 200 nucleotides, and then they are joined to each other by the DNA ligase. So once there is unwinding of these two strands, there will be one strand formation in the continuous way, and the other will not form in a continuous, it will form in the discontinuous will form. It will initially form some fragments, and some fragments, and these fragments will be joined by the DNA ligase. Okay, so that is called as Okazaki fragments, which happen in the lagging strand. Okay, so there is a leading strand and the lagging strand. Okay, so you will have the parent DNA, which is helical. Then there will be unwinding of the strand. Each strand will get one new strand, but one will be the form leading strand, other will be the lagging strand. In the leading strand, there is a continuous activity, while in the lagging strand, it is in a discontinuous format. Synthesis is in the discontinuous format. You get a multiple primers there, and you get the Okazaki fragments, and it takes time also. Okay, and ultimately there are ligases which will join these strands. Okay, so leading and lagging strands are not pre-existing strands. They are not separate DNA molecules. They are newly synthesized strands formed on the opposite template strands at the replication fork. What does the RNA primer does? It is synthesized by the primase. It is required for DNA polymerase to start its action. Later, it is removed and replaced. Okay. So DNA polymerase cannot act on its own. Then there is a DNA ligase. It will join the Okazaki fragments and it forms the phosphodiesterase bond. Okay. Coming to the DNA repair mechanism. Okay, so in these DNA repair mechanisms, there is NER, that is nucleotide excision repair, and function is that it UV damage and thymine dimers. Then mismatch repair, it corrects the replication errors, and base excision repair, small base damage. Okay, so these are the mechanisms which are happening. Here there can be a question, xeroderma pigmentation, where there is a NER defect. So this mechanism, DNA repair mechanism, NER, is defective. So that leads to the xeroderma pigmentism. Here feature is UV sensitivity, skin cancer, etc. Then ataxia telangiectasia, here ATM gene is responsible. Ataxia plus cancer can happen. In Fanconi's, DNA cross-linking repair is defect and bone marrow failure can happen. In Bloom syndrome, helicase is defective. Cancer risk is there. In Lynch syndrome, which we see in gynec, which is a mismatch repair gene is defective, and it leads to the colon cancer, even corpus cancer also. So mutation is a change in the base sequence, and cause may be replication error or repair defect. Okay, so just to revise, DNA synthesis happens in the 5' 3' direction only. Polymerase needs DNA primer. Leading is a continuous, while lagging strand is a discontinuous synthesis. Okazaki fragments are joined further by the ligase. Then helicase will cause unwinding. Topoisomerase will relieve the tension, and this NER xeroderma pigmentism is because of the NER defect, and it causes UV sensitivity. Okay, So DNA polymerase reads the template in 3-5' direction and synthesizes the new strand in the 5-3' direction, because nucleotides can only be added to 3-hydroxyl end. Okay. Then there is a concept of telomere and telomerase. Okay. So telomeres are the repetitive DNA sequences at the chromosomal ends. So each chromosome has got end, and there are certain sequences which are fixed, and their role is very, very important because they protect the chromosome from the degradation and the fusion. So end of the chromosome is very important. Okay. So the repetitive DNA sequences at the chromosomal ends, they are responsible for protecting the chromosome from degradation and fusion. So in human, this sequence is a tag, TAG. Remember, 2T, 1A and triple G. Okay. So TAG, TTAGGG. Okay. So tag, if you remember, there are 2 T's and triple G. Okay. So end replication problem can be there. So DNA synthesis happens only in 5-3' direction that we have seen. Then when there is a problem, it cannot replicate extreme 5' end of the new strand. So when there is a problem, the 5' end will not be replicated. So result will be loss of 50 to 300 nucleotides per division. Okay. So DNA polymerase cannot fill the terminal gap. Okay. So what happens without telomerase? So here comes the role of telomeres. Okay. So these telomerases, what exactly they do? So terminal RNA primer is removed. That leaves the gap. No DNA synthesis is possible. That leads to the shortening of the DNA. Then exonucleases action, there is a further degradation that will lead to the chromosome instability. Okay. So that will lead to the end-to-end fusion, DNA damage and cell death. So if there are no telomerases, then this can happen. There can be chromosome instability because of the end-to-end fusion, DNA damage and cell death. And this is prevented by these telomerases. Okay. So what you should understand, with each cell division, telomeres shorten. That leads to the aging and genomic instability. Okay. Then telomerases will come into picture. Enzyme type is a reverse transcriptase. Template has got intrinsic RNA template. Action is it adds repeat to 3' end of the DNA, and function it prevents telomere shortening. So whatever the shortening should have happened, that is prevented by these telomerase. Okay. And the enzyme type is a reverse transcriptase. You just remember these certain things. It reverse transcriptase activity is there. So telomerase binds to 3' end. It uses its RNA template, add telomeric repeats, that is TTAGGG, which is at the end. So DNA polymerase completes complementary strand. So finally, this DNA polymerase is able to complete the complementary strand. Otherwise, it would not have completed. Okay. So telomerase is reverse transcriptase that rebuilds the chromosome ends. This concept you should remember. Okay. So clinical correlations. So aging, what happens? Older individuals, there is a shorter telomeres. So that leads to the decreased telomerase activity and resulting in cell senescence. So that this happens in the aging. There is a cell senescence because of the decreased telomerase activity. What happens in the cancer? This telomerase activity is increased. That prevents the shortening, and there is a cell immortality. So more and more cells. Okay. So persistent telomerase is the hallmark for the cancer. Okay. So aplastic anemia, there is a premature telomere shortening, and disc keratosis congenita, there is telomerase defect. Okay. Then what about the hydrolysis of the DNA? Okay. Which enzymes are responsible? Nucleases. Okay. There are two types of nucleases, exonucleases and the endonucleases. Exonucleases will cut from the end, while endonucleases will cut the DNA within. Okay. Then there are restriction endonucleases. Now these restriction endonucleases, also called as molecular scissors, their action is to cut at a very specific sequence. So they will cut at a very specific sequence, not randomly. So this is used in the genetic engineering. Okay. What are the inhibitors of DNA replication? Ciprofloxacin. Here there can be question. Okay. Ciprofloxacin inhibits DNA gyrase. Nalidixic acid, again DNA gyrase, and novobiocin DNA gyrase. Okay. So these are the antibacterial drugs which will inhibit the DNA. Then anti-cancer drugs. Etoposide will act on the topoisomerase. Okay, doxorubicin will also inhibit topoisomerase. Adriamycin will also inhibit topoisomerase. These are all questions which are asked, how these drugs are acting. Then 6-mercaptopurine, DNA polymerase inhibition. Okay, 5-fluorouracil, thymidylate synthase inhibitor. Okay, then fluoroquinolones will inhibit the DNA gyrase inhibitors. Then 5-fluorouracil will decrease thymidine, leading to decreased DNA synthesis. Etoposide will prevent the DNA unwinding. Okay. So telomere, chromosome end protection, that is the job. Problem is that end replication gap can be the problem. So telomerase activity is through reverse transcriptase. Aging reduces the telomerase activity. Cancer, there is an increased telomerase activity leading to immortality. Exonucleases, the end cutting is done by the exonucleases. Endonucleases will do the internal cutting, and 5-fluorouracil will thymidylate synthase inhibitor. Okay, this is the 5-fluorouracil. Okay, doctor, we have few questions. Can you please go ahead with the questions, doctor?
Yes, sir. DNA polymerase is unable to replicate the extreme end of the lagging strand because it lacks proofreading ability. It cannot synthesize in 3' to 5' direction. It requires RNA primer. It cannot add nucleotides to 5' and it lacks helicase activity. So it requires RNA primer.
Yes, correct. That is the, so it cannot add nucleotides to this. Sorry,
Okay.
DNA polymerase is unable to replicate the extreme end because
Extreme.
it cannot add nucleotides to 5' end. This is what is the answer. It cannot add nucleotides to 5' end. It
It
requires
adds to the
RNA primer
3'.
for beginning this transcription. Okay.
Hmm. Okay.
Extreme end, we are dealing with the extreme end. We have just seen, no, extreme end is because it cannot
Yes,
add
sir.
nucleotides to 5' and there comes the role of the telomerases. Okay, so DNA
Telomerase.
polymerase adds
Yes.
nucleotides only to 3' hydroxyl end, so the terminal 5' end cannot be filled. Okay, and that is why the
Hmm.
telomere shortening is there.
Yes,
Next.
sir. Function of telomere. What is the primary function of telomeres? DNA replication, protein synthesis, chromosome protection, transcription regulation, RNA splicing. Sir, chromosome protection. Option
Correct.
C.
Okay, especially the ends. Okay, so it prevents the
Hmm.
DNA degradation and end-to-end fusion. Next.
Telomerase is best described as DNA-dependent DNA polymerase, RNA-dependent RNA polymerase, RNA-dependent DNA polymerase, DNA ligase, endonuclease. Sir, RNA-dependent DNA polymerase.
Correct, because it is a reverse transcriptase. Okay, so it
Reverse
will be
transcript.
RNA dependent DNA polymerase. RNA dependent DNA polymerase. So acts like a reverse transcriptase using RNA template, and it has its own template. Oh, okay. Next,
Telomerase acts on which part of the DNA? Sir, 3' and of lagging strand.
is it 3' end of
3' and
telomere?
3' and
Okay,
telomeres. Yes, yes,
telomere.
sir.
So it adds repetitive sequences tag. Okay.
Yeah.
Next.
Shortening of telomeres is associated with increased DNA synthesis, cell immortality, aging, and senescence. Sir, option C, aging and senescence.
Yes, correct. Shortening of telomeres will lead to aging and senescence. Next.
Which of the following is characteristic of most cancer cells? Reduced DNA synthesis, short telomeres, increased telomerase activity, absence of replication, increased exonuclease activity. Sir, option is C, increased telomerase activity.
Correct.
An
Yes,
enzyme that removes nucleotides from the ends of the DNA is, sir, option B, exonuclease.
correct. Exonucleases. Okay. So exo will act at the end, endo will act internal. Next.
Restriction endonucleases are best described as random DNA cutters, RNA cutters, sequence-specific DNA cutters, DNA synthesizer, repair enzyme. Sir, they are the repair enzymes. Option E.
Option
E.
Restriction. Endonuclease.
Restriction
Endonucleases,
endonucleases.
that means they will cut.
We
But
cut
what is the
inside.
exact way of cutting? They don't cut
Sequence
randomly.
specific DNA cutters.
Yeah,
Yes,
they
sir.
will cut
Option C.
sequence specific. Okay, that is very important. No, sequence
Hmm.
specific DNA cutters. They are molecular scissors, and that has got a role in the engineering. No, we have
Yes,
seen
sir.
biomedical engineering, genetic engineering. It has got role. Next,
Which drug inhibits thymidylate for synthesis? Synthase. Sir, option D, 5-fluorouracil.
yes. Okay, 5-fluorouracil decrease thymidine, decrease DNA synthesis. Next,
Ciprofloxacin acts by inhibiting, sir, option A, DNA polymerase. Ciprofloxacin,
DNA gyrase, doctor.
novobiocin, and
DNA gyrase, not
DNA
polymerase.
polymerase.
Okay.
Okay, all the three of these.
Okay, so DNA polymerase cannot fill 5' end. Telomere is protective for chromosome ends. Telomerase, RNA-dependent DNA polymerase. Aging is because of the decreased telomerase activity. Cancer is because of the increased telomerase activity. Exonucleases will cut the ends. Restriction enzyme are sequence specific. Then 5-fluorouracil will lead to the decrease in thymidine, and ciprofloxacin will act on the DNA kinase and it will inhibit the DNA kinase. So this finishes the DNA structure. Okay, thank you.